I was really impressed that on the first day, we had 100 participants signed up for the pain meeting from 20 different countries. And we also had about a 50-50 balance between men and women attending, which we really tried hard to plan in terms of inviting people that would represent a variety of fields from early preclinical to advanced clinical, and to have those who are early in their career, later in their career, and have a balance of men and women.
Yeah, especially in our speakers. We have all areas of disciplines represented.
Yeah.
We have both genders or male and female speakers represented, and we have junior faculty, some postdocs presenting, I think even a student presenting, in addition to giving short talks at the end of our sessions.
Yeah, the other thing that we go to too many pain meetings, to be really honest, and what was particularly special so far is that the hope was that people would present new unpublished data, and by and large that's what happened. The talks, people are incredibly kept to time, which is almost unheard of, but there was an enormous amount of new ideas, not only background in how pain is generated, both in preclinical studies, but also some beautiful introductory, great talk from Irene Tracy, looking at human brain imaging, trying to get a handle on what's happening in the human brain, that the idea that she made the case that there's no one pain area. That led off the whole meeting, but then it went into very detailed discussion of new targets. But even then, and Bill can talk more about this, the perspectives on taking molecules into the clinic. There was even an interesting discussion as to whether about animals versus translation and to what extent they translated. So we covered almost everything, and it was just all positive.
You know, one of the things I think is really important is that too many people have had the impression that there's nothing in the pipeline, that nobody is doing research in the area of pain that will overcome our dependence upon aspirin and morphine, as an example. But what I've come away from here is that there is a lot of development that is going on, some of which may be nearer term with some new drugs that may be on the market in as few as 3-4 years from now, but also a lot of new targets that are being evaluated now. So it's not just opioids or NSAIDs, it's a whole variety of different things that can address pain in general or specific types of pain for certain types of patients.
Yeah, I think it was also good we had a session representing NIH HEAL initiative, and had many members from the different agencies at NIH talking about their efforts, the publicly made resources, just making them available to investigators to help their own research endeavors. I thought that was a really helpful session.
I think so too. I think most of us working in the field have heard about the HEAL initiative. It's put an extra $1 billion or almost $1 billion into areas of analgesic research in the last 2 years, or also for treatment of opioid addiction. And while we focused on the analgesic field here, what I've seen is that there are a lot of academic labs that now have financing. A lot of small companies that had languished are now getting some financing. And what was announced at this conference that maybe a lot of people were not aware of is that the NIH, through various institutes, have set up a new screening program at the preclinical level. It's called the PSP, the Preclinical Screening Program, where anybody from anywhere in the world can submit new compounds, and they will run them through a battery of standardized tests and report back at no charge, report back standardized results to indicate whether this might be something that should advance to the next stage. And if you do have the opportunity to advance to the next stage, the NIH has other mechanisms to take over. And one of the other areas that they discussed was a new program called EpicNet, where on a competitive basis, a company or individuals can submit compounds that already have an IND submitted or that are IND ready, as they call them, so that they are ready for clinical trials. What EpicNet will do is provide resources for a phase 2 clinical trial, a proof of concept clinical trial. And that's where most compounds go to die.
Yeah. Yeah.
If we don't get beyond phase 2.
The valley of death.
It's the valley of death.
Valley of death.
So NIH is going to help us, and that's at no charge, no fee, that they will work with the sponsor to design an appropriate clinical trial, And they have a clinical trial network set up across the country to be able to do these trials.
Yeah. The other thing that came out of the blue, so Chaz Bountra, formerly from industry, he was for years doing pain research at Glaxo in the UK. He's now at Oxford, and he presented a story of which I was completely unaware, that they have a program now of developing and identifying new ligands, new potential, call them drugs. We don't know if they're drugs yet, but that they're designed to target specific receptors that have been characterized from human cell lines, human proteins. And what was particularly remarkable is that they are making these compounds, and there's about 70, I think he said now, and they've identified targets available to anybody in the world. No intellectual property involved, which is almost unheard of, because pharma has lots of compounds, but we can't get them, or you sign your life away in order to get them. And this was an eye-opener on a personal level, to be able to get a hold of some interesting compounds that might be targeting something that you have reason to believe is important. That was an example of the breadth of what was covered, from fundamental science all the way to, hey, here's an opportunity for you to take something either to the clinic or to do your own work, both for industry and for basic scientists in the lab. That doesn't happen at most meetings.
Yeah, we also had some really interesting technologies presented, especially whether for monitoring or observing animal models and more visual assays and automated big data kind of collection systems, learning algorithms to determine just facial recognition of pain in animal models. These kind of studies were very cool, and along with the work to suppress pain responses by using really innovative new tools that I think are going clinically as well.
Yes.
Some of them are in the clinic already. Some optogenetic tools to inhibit pathways wirelessly. It's early days, it's cutting edge, so the technology is starting to bridge. I mean, one of the big problems we've always said about, and it came up at the meeting in an interesting way, someone said, cancer research is easy because you have less concern, you have a concern, but the drugs that you develop are generally toxic because they're trying to kill a tumor, and so inevitably you're going to get toxicity, and there's how much you're willing to tolerate is much more than with a pain drug. You're not going to tolerate miserable side effects. And the other thing is placebos don't work for cancer. This is a huge barrier that we have to face. Placebos are very effective in some conditions, and to get something through the FDA. So the approach is to use different methods, better biomarkers to assay both in animals and humans, is something that also came up and is encouraging. We're not there yet, but the direction is, there's a kind of a common theme that people are going after, which is good news.
You know, taking up on that, I was impressed by the second or third talk on the first day by Lauren Heathcote, who is a fairly young faculty member at Stanford. And she pointed out in particular that we have fewer drugs to treat pain in animals than to treat pain in humans. And you know, as I thought about it.
No, we have more drugs to treat pain in animals, don't we?
No, actually.
There's fewer in animals?
Fewer.
Oh, okay.
Because, as an example, a lot of people may not be aware of this. NSAIDs are incredibly toxic to dogs and to cats.
Cats, yeah.
And if you take something very common like ibuprofen or naproxen and give therapeutic doses, analgesic or anti-inflammatory doses to a dog, that dog probably is going to have a major GI bleed within 3 to 4 days. Mm-hmm. And that could be lethal for that animal. We know that NSAIDs can cause a lot of toxicity in humans. Why humans are more resilient to that than animals has really never been resolved. And you know, even then, some animals would respond and other animals would not. But it limits the types of drugs that can be used in animals. Even opioids. Opioids you can use in some animals, but dogs will have much more sedation than humans. Horses become excited. Cats also become manic in some cases. Yeah. Feline mania is...
Especially under opiates.
With opiates, yes, exactly. So I think if we plan a conference like this in the future, we'll have to include some veterinarians working in the area of pain medicine.
Although we did see today an example of a horse being treated very effectively. Yeah, no, so that was pretty incredible.
That was pretty interesting. Going back to the Lauren Heathcote, now, I was the individual who introduced her to the group initially when we were looking for names, because I heard her speak at the North American Pain School way outside of Montreal where I grew up. And she gave this talk. She was a postdoc at Stanford at the time, working with Laura Simons, trained in Britain, where she did a lot of imaging work in the brain of infants. And I thought that that's what she would talk about, but that's what we invited her to talk about, but she didn't, because she knew that it was supposed to be unpublished story. And what she talked about, it led to great discussion, the fact that the messages that are sent to children, particularly when they are being treated with a new drug or an old drug, and there are side effects. And so if you tell the kid, well, yeah, you know, there'll be side effects, but don't worry about it, blah, 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 blah. As opposed to, if you get those side effects, that means the drug is working. And what they found is that if the kind of message you send to the kid will completely change the likelihood that not only you get better responses, the willingness of the kid to accept the drug. So what came across at the meeting was pain is in the brain. Psychology influences it. That's why placebos work. But her talk was so convincing, and I think it was a good illustration of young folks coming up and bringing some new thoughts to the field. It was great.
Yeah.
You know, get back to what Laura just said about big animals also being shown at this meeting. One of the major complaints that I've heard over many years is that animal pharmacology doesn't translate to efficacy in humans.
We
We
heard that at this meeting.
We heard it time and time again. But I think one of the things that has come out is that there are newer animal models, less biased models, more objective models that can be used. And we can also go from experimental models of pain in animals to pathological models of pain. And that's what we saw in one of those videos today, where a horse was treated and had a remarkable response to a new type of drug, new mechanism of action. Horse with osteoarthritis that was limping, we saw that limp, and after treatment with the drug for several days, the horse was literally running around the corral.
Yeah.
So I think the take-home message is that there is a lot of new research that's going on in pain medicine. And here at Keystone, we're hearing some of those breakthroughs right now. And I think we're going to see a lot of those translate into new things, new targets in the clinics in the future. And some of them could, in fact, replace many of the drugs that we have today, NSAIDs and opioids, all of which have adverse side effects. But these may end up being some targeted therapies, some that may work better in certain types of pain, like neuropathic pain or in inflammatory pain. But I think, at least what I concentrate on in the clinical research that I do, is finding drugs that are more effective with fewer side effects and without addiction liability. And I think we're going in that direction.
Yeah.
I think the other message that came through, there's something difficult about pain research, and the way I've talked about it in the past is that you don't die of pain. You may die in pain, but you don't die of pain. And the significance of that is that, by and large, the only source of money for pain research, if you're going to do discovery work or that kind of work, is the government. Pharma sometimes will give you some money, but it's usually to test their drug, which is boring. You don't want to test their drug. There are very small little enterprises focused on specific types of pain, but there's nothing equivalent to the American Heart Association or American Cancer Society or multiple sclerosis, because pain is not seen as a disease. That was another message that was brought to the audience, is that in many ways chronic pain is a disease of the nervous system. I don't think that's going to necessarily convince someone to start a foundation that will fund it. But that was the significance also of the HEAL initiative, where the government finally said, we need to step up to the plate. There was a wonderful talk from Chris Beasley, who represents the Chronic Pain Research Alliance. She's not a scientist, but she is an incredible advocate for pain research, as well as for teaching the world about pain. And she's shown numbers that I've seen many times before, where the money spent on pain and the incidence of people with pain far exceeds that of, say, cancer, diabetes, heart, all together. Now, she's preaching to the converted here, to the choir, and that's a bit of a problem, and the choir is really outside. It would be wonderful to get that message farther out. The HEAL initiative is government's effort to make a difference. But her talk, following upon Irene Tracy's talk about brain imaging, was just an interesting juxtaposition of where the science is going in humans and where the problem is in the human. You don't see that at other pain meetings. This was
And,
special.
you know, there was lots of discussion that came out of these presentations, and you initiated one bit of discussion that was really important after Chris Feasley's talk, which is bringing scientists to see pain patients sometime. You saw that when you were a young scientist. I've seen that
when
too.
I was a young scientist?
Well, all right.
Never mind.
We need
that
to
was a
do.
takeaway for me too.
Yeah.
I like that concept. I thought about how clinicians follow and shadow and have these rounds, and the PhD scientist is not selected for their empathy. They're selected for their focus, and this is something that I think we should build into our training programs. That was something I took away from it, that the PhD scientist needs to understand the problem.
No, it's and it's very doable, although we're starting to reach barriers. I send my students and fellows to the pain clinic. I did that when I was a young scientist, to just sit, listen to the patients, find out what works, what doesn't work. Most things don't work. There's traditional drugs. What's happening now is because of HIPAA, there is real concern to allow students and postdocs to even go and spend time with the patients. So these are barriers that are so annoying, they're bureaucratic, but it is absolutely critical that these students, these young scientists, learn that a mouse and a rat are not human. There's a big difference. And that came out, so that was also good.
Yeah.
We should talk a little bit about the issue of the structural organization. Everything was wonderful, really remarkably organized. Talk's great, but there was a parallel meeting going on, and speaking as somebody who works in somatosensation, that's the meeting that was next door. We could do it better in the future to find a way to get more bridging, because I can guarantee you there were talks that I wanted to hear, and there were talks that they wanted to hear, and there's got to be a way to do that. And it was sort of driving me nuts that it was inappropriate for me to leave our group to go hear something. But we could work on that in the future, and we should make notes of that.
You know, there was still a bit of cross-fertilization going on, because we did share breakfast and lunches and so on. I actually had been looking forward to meeting one of the people who was talking at Somatosensation, and I knew I couldn't attend his talk, but fortunately he was on the same bus from the airport that I was on, and so we had a chance to chat a little bit.
And there's the posters.
And the posters,
are
that's
joined,
right.
so that's helpful.
Yeah, the posters were excellent. Yeah, and there was enough time and good time to discuss, so fair enough.
Yeah. I think I'm in the wrap-up of our meeting we haven't had yet. I'm looking forward to our cannabis session. So yeah, we wanted to bring this this topic, and we wanted to have it as a discussion basis at the end, and so we've left time for that. When we started planning this meeting, I guess almost 2 years ago, we said, well, let's wait and fill these talks because we need to see all these clinical studies that are obviously going to come through in the next year that are going to tell us about cannabis and pain, and CB1, CB2, and pain.
Clinical studies take a long time.
And
And a long time to conduct.
Yeah, and so we still didn't have very much, and so and we're also going to discuss. Really, there's a lot of question as to whether this is a useful approach, and I'm looking forward to that discussion.
But I think Laura you brought up a good point during one of the discussions today, which is we need to ask patients when they join clinical trials ahead of time, what are the prior drugs that you've been using, and we have to think. Is cannabis a drug? Well, in some states it's not. In fact, it's still a Schedule I agent. And so they may be reluctant to tell you about that.
Yeah.
But that could be a contributor to the overall efficacy and the side effects of what we're looking at in terms of new drugs.
And interactions.
And that's right.
Absolutely.
One thing that we haven't got into, which is worth thinking of when we go to the future, because I was involved in the National Pain Strategy government-sponsored effort to try to define priorities. And we divided up working groups, but disparities was one major group, because you're talking socioeconomic, racial, educational, whatever. People respond, and age, of course. Huge difference from pediatric. For a variety of reasons, we tend to be a little homogeneous in the populations that we study, either preclinically and even clinically for a variety of reasons. The brain changes, and we know that, how we learn, the context in which we experience injuries. So that's something that it's a little more focused, perhaps, but it's something that I think we should consider in the future, which we tended to ignore here.
You know, that reminds me of one other thing that the government is doing on a national basis right now, which is to encourage more research into biomarkers that will be predictive of whether a patient is going to respond to a particular drug or whether they have pain to begin with, whether that pain is resolved. And there's a new program worth $40 million from the Office of the Director that will fund research to determine which patients are going to transition from acute to chronic pain and which patients have either susceptibility or resilience toward that transition, so that you can plan differently in terms of how you start to work with a patient and even maybe which drugs you choose to treat pain for those patients.
But just for the record, one of the most, because that was certainly one of the working groups, transition to from acute to chronic pain, and exactly what you talked about was brought up, but there was an interesting discussion, is who, for all we know, there is no transition, that you are chronic from the get-go, that the problem is there, and that we have some artificial definition of 3 months is chronic pain. That's an totally artificial definition. It could be 1 week when the transition occurs, or it could be you're going to be a chronic pain patient from the get-go after surgery. Who knows? So there are plenty of questions left for us to do. I would also want to talk about a somewhat intense but good discussion that continued outside the sessions. What is the best approach to developing a new drug? The two sides. One is to try to understand the normal organization of the brain and the pathological consequences of injury and how that affects pain processing. That doesn't necessarily say where the best drug is going to work. And we didn't really get into that, but we sort of reached it from the outside. I mean, many people believe the best targeting is in the periphery. Well, most of what we talked about was what's going on in the brain as a consequence of injury and in the setting of chronic pain. That doesn't mean to say that that's the best target. And I think at some point we need to make it clear to the audience, because some people were upset and said, well, you're wasting your time studying that. It's like saying, well, so I shouldn't understand the normal organization of the body? That's ridiculous. So there were some interesting things that we didn't have time to, but I think if we could set up some really interesting debates in the future. It could be kind of fun.
But I do appreciate that throughout the meeting, I feel that the discussion has all been very constructive.
Yeah.
It's been a good discussion. It's not been, I don't feel like there's been an attack or defense. It's been a very good meeting from that sense, and I really appreciate that.
Cross-fertilization, I think, is really important with a meeting
absolutely.
like this. Bringing basic scientists and clinical scientists together, we all have a common objective, which is to find better ways to treat pain, better ways to diagnose pain, better ways to measure pain. And with a dialogue like this, I think that we've made an advance with this meeting.
Yeah.
I think the one thing that we knew was going to be a problem right from the get-go, and the other group actually had some ideas as to how to deal with it, and it's something that happens all the time. The fact is that students and postdocs tend to be pretty hesitant to get up and ask questions, and you know they have questions, but they're afraid it's going to be a bad question or a dumb question. There's no such thing as dumb question. You tell them it doesn't matter. And so they had these ideas that they were going to give drink tickets out to any student, and then we were told, no, that's not going to happen at this meeting. Okay, so then they came up with the idea they had the golden ticket, which if a postdoc or a student asked a question, they get a free interaction with any faculty member they want. The person has to interact with them. Kind of amusing, but it is a way to try to get the younger folks to be more engaged, because it is true most of the questions came from the more senior members of the audience. That happens all the time, but you really would like to engage. There were a few exceptions. There were a few, but not as many as we would like.
I think that responsibility should lie on us as well. I think it's up to us to go to their posters and go and interject ourselves.
Yeah, the posters, I looked carefully, and there were a lot of really interesting ones.
Yeah.
There were very few where there was sort of somebody standing there praying that somebody would come, like at the Poor Society for Neuroscience, and you always see this young student who's sitting there, and she's been there or he's been there for 3 hours, and nobody, and sometimes I'll walk up to them and have a discussion, and something, I have no idea what the poster's about, but you just feel badly. You feel bad, excuse me. That wasn't the case here. There was a lot of activity at pretty much every poster.
Yeah.
It was good. That was good.
Well, I thought it gave us a chance to see what was going on in the somatosensation sessions. That was like the one opportunity we really had to go, oh, it's maybe their talk is now reflected in part of the posters. So that's what I've been looking for in the poster sessions. And then, of course, our own posters. I had some very good discussions. I'm looking for postdocs, so I'm like scouting. So tell me, are you a student? How many years?
You know, I sat next to somebody at breakfast this morning who is a postdoc overseas and is looking for some new positions. Maybe I can introduce her to you later on.
That would be fantastic.
But that sort of interaction is important too.
Yeah.
20 countries, and a
Yeah.
lot of the people coming from outside the United States are younger faculty or students or postdocs.
And you know, what was really great with the meeting is that we did get funding. We did get an NIH fellowship from NCCIH, and we were able to offer, how many, 7 or 8 fellowships to help with student travel to come to the meeting and trainee travel. So a number of people came up and thanked us for making it possible for them, especially. And it seemed like it was a bit on a sliding scale, that it was a need-based, because some expressed that they felt there was no way they could have done any part of this without the contributions. So I thought that was really great.
Yeah.
For me, it was the posters. I had some great interactions, and I was actually taking notes or typing things quickly on my phone, since you're not allowed to take photographs, which I get. But more methodologically, there were some really novel approaches that were used. I wasn't aware of them. They're relevant to what we're doing. Some results that differed from what I certainly believed, and they were presented by students and postdocs and very knowledgeably. So that was, in a very practical way, very useful. And in fact, I sent my own postdoc. I said, you got to go see that number so-and-so. It's very cool and it's relevant. So this will continue, because there's a little more breadth. There's so much published these days, you can't keep up anymore. Why do I go to meetings? I go to meetings to mostly posters. At the neuroscience meeting, I don't go to talks. I go to posters, because then I can find out and talk to people, find out what they're thinking about, what their ideas are, and then really get some cutting edge stuff. And as I said, cutting edge, because most of what was presented here has not yet been published. And that's really unusual.
You know, I remember 2 of the posters from last night that fall into an area that I'm particularly interested in, which is chemotherapy-induced peripheral neuropathy. This is an area where we don't have any approved drugs, and where we know that the patients who are suffering from CIPN either have cancer or they've been treated for cancer and they have effects that last, in some cases, for the rest of their life. So finding students who are now working on CIPN with novel ways of being able to look at this, I found refreshing. And I had a chance to talk to both of the groups of people last night and ask them a few questions that at least I've thought about that they now can think about too, and that'll help to focus their research.
There was one specific one related to that, because we study CIPN, and it had to do with an oxaliplatin platinum poster where they used a different dose from what we use, and they got cold hypersensitivity, which is very characteristic in the animal and in the human model. It's a huge problem in humans, but they concluded, based on all their work, that it is not TRPM8 mediated. And then the question is, well, and it's not A1 mediated, it's getting into the weeds here, but the question is, what's the target? And it's not at all clear what's mediating, what is basically one of the major reasons why patients will stop taking the drug. Because Prematurely, no, absolutely. They'll stop taking it because, the pain just makes it impossible. Well, it's sort of like an opiate, a patient on opiates who's getting so severely constipated that they just say, I can't take it. And that's all the reasons we know we're preaching to the choir here again, why we need something different. And there were a lot of good ideas and some promising hints. It was 2 years ago at a meeting, what was it? ACMP meeting, which is a meeting I normally don't go to. And they had a pain session and Laura invited me. And we talked about it, and she brought up the idea, and I have to say my eyeballs kind of rolled because, oh yeah, just what we need, another pain meeting at 12,000 feet, no, 9,000 feet. And I said, well, it's 2 years from now, sure. And
That was my hook.
that was,
That it's 2 years from now,
it's
Allan.
2 that's right, it was 2 years
You
from now.
can say yes.
That's right. You haven't committed yet. There's nothing to worry about. And then slowly, and then Bill came in, and we started having discussions, and it evolved with the intent of being a very different meeting from the ones I used to go to.
Yeah.
I'm used to going to, in that it was broad, but still focused, and went from discovery all the way up to clinical trial, introduced aspects of the patient. I don't think we had that idea necessarily when it began. My guess is it would have been another discovery, preclinical type meeting. That's what I anticipated, and it didn't turn out that way. So kudos to you.
Thank you.
I think we had a good balance.
I think we did have a good balance, and that was the point of bringing the group together, I think, to get the different perspectives. I mean, I'm a card-carrying pharmacologist, and I tend to look at a lot of receptors and signaling assays, and I know Bill has a tremendous experience in preclinical and clinical drug development. Of course, your absolute expertise in pain mechanisms and neurobiology. I think it was, and I think we were all really dedicated to make it different than the typical meetings that we go to, and to educate ourselves in the different aspects as well. So
I think we did it.
yeah.
Yeah.